So when I started working in Liz Blackburn's laboratory, it was very clear that the crucial question was how are chromosomes maintained. And so we set out to test the idea that maybe there is a new enzyme in the cell which people hadn't known about before, which would simply recognize the end of a chromosome and add sequences onto the end of the chromosome, essentially lengthen the chromosome. And the very important thing that Liz had found when she was a postdoctoral fellow with Joe Gall is that the telomeres themselves are very simple DNA sequences. So in this organism Tetrahymena, where telomeres had first been identified, a Tetrahymena cell has 10,000 chromosomes, and so it's much easier to identify telomeres than in a human cell, which has only about 100. So the telomere sequence is a very simple sequence. It's T, T, G, 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 repeated hundreds of times. T, T, G, 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 T, T, G, 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 hundreds and hundreds of times. And so what we looked for was if we take such a sequence or a different sequence, can we find an enzyme which will just make that sequence?
So the first really clear, definitive evidence as we look at it now that there really was an enzyme that we came to call telomerase came from an experiment that Carol had done very late in December in 1984, and she had got the result and she showed it to me the next day. And I looked at this sequence and she was very thrilled because she clearly recognized the significance of this. And I saw this and I looked at this thing and I just thought, whoa, this is really big. In fact, it's interesting why I was so confident at the time that this was a really important result. And I think maybe we'd been sort of building to it in lots of ways. There had been a lot of different lines of evidence from the way telomeres had been behaving in vivo and, you know, the preliminary experiments and then more and more. So in some ways, it was sort of gelling, I think, even at that point in our minds.
We felt that it because the telomeres themselves are really conserved throughout evolution. That is, the simple tandem repeat is present in yeast, it's present in humans, it's present in mice, in Tetrahymena, that whatever we found about the Tetrahymena enzyme would be true in all organisms. So when I first developed the gel that told me that there was a 6-base repeating pattern, that there was this enzyme, I was very excited about that. It was actually in December of 1984. But again, my first thought was, okay, what else could be going on here? And it was about a year later when we did an experiment, which was really the converse of the experiment that Liz Blackburn and Jack Szostak had done. What they had done was to take a Tetrahymena chromosome and put it into a yeast cell and find that yeast telomeres were added onto that chromosome. So what we did was to take a DNA oligonucleotide, which was a yeast sequence DNA oligonucleotide, and put it into a Tetrahymena extract, and we found now that the Tetrahymena sequences were added on to the end, saying that this really was something specific. There was something in the Tetrahymena extract that knew how to synthesize Tetrahymena telomeres even when given a yeast chromosome end to elongate. And it was that experiment and the details of how the patterns looked on the gels that absolutely convinced me.
And I remember looking at this and just having this very strong reaction. This is it. This is the real thing. That this really was the first kind of clear evidence that yes, this thing that had been suspected for so many different reasons, that it really might actually exist.
That night I went home and danced, because then I thought it really is something new. And that was about a year after the initial discovery.